The first sketch is to show how saliva is secreted and show what is in the saliva. So here's a salivary gland in the head of an animal. And if we zoom in on the salivary gland, we've got goblet cells that are secreting vesicles of mucus. So these would have mucus inside that's been made, protein core and sugar chain side links, and those fuse with the membrane through mechanisms we've talked about, and we get mucus. Similarly, enzymes, proteins, are manufactured and packaged, often tightly packed into granules. They're sort of crystal-like. And those also, through mechanisms we've talked about, they fuse with the membrane and they release, in this case, lysozyme in saliva. Then to produce the fluid component, we have cells in the epithelial layer, and the NKCC transporter moves one sodium, one potassium, that's the N and the K, one sodium, one potassium, and two chlorides, that's the CC. It's a secondary active transporter that is powered by sodium moving in, and that brings the potassium and the chlorides with it. Of course, the potassiums are recycled, and the sodiums are pumped back out through the sodium-potassium pump. The chlorides inside the cell now are in higher concentration inside than outside, and so with a chloride channel, the chlorides move out. That moves net negative charge into the fluid in the salivary gland, and the tight junctions between these cells here are leaky to sodium. They function like a sodium channel. They have charges and gaps that just fit the hydration shell of a sodium, and then water may be moving through leaky tight junctions or through aquaporins through the cells. And the water, of course, is forming hydration shells around these sodiums and the chlorides. Put that together and we get some mucus, we get some enzyme, lysozyme, that breaks down, and amylase that breaks down some sugars, and then the salty fluid. The second sketch was some elements of structure of the stomach and acid and chloride, and then pepsinogen. Again, there are goblet cells producing mucus, and that forms a mucus layer over the interior of the cell. Inside that mucus layer is being secreted acid, and you could do this with any proton pump. There are multiple proton pumps. The one that happens to be used is a proton pump that exchanges a potassium for a proton. That keeps the charge balance there. The potassiums, of course, leak back out, so that means you get more positives outside than in, and so chlorides will follow. So we call it hydrochloric acid, but in fact these protons in these chlorides were never together. They weren't ever an acid. They were separately pumped as protons through this proton pump or proton potassium active exchanger and a separate chloride. Pepsinogen is a precursor for an enzyme. It's packaged in vesicles. It's in an inactive form, a zymogen it's called, an inactive form, because an active form would be quite dangerous inside the cell. It would easily start digesting whatever's nearby. So it's put in as inactive form, and these protons cause part of it to be cleaved off. There's a acid-sensitive region, so protons will slowly break pepsinogen into pepsin, and then once you have some pepsin, pepsinogen actually binds to pepsinogens and cleaves off part of it to form more active pepsin. So you need a little bit of acid to get it started, and then from then on, the pepsins will do it. That gives us an acidic stomach. Better to sketch it separately with protons and chloride separately because they're never together. Next one is sketch showing secretion of enzymes and buffer in the pancreas. So here is pancreas epithelial cells. Here is the lumen. This is going to go into the digestive system. And over here, we're seeing, here is the lumen. So this is the side that is going to be sending stuff to the intestine. And cells in the pancreas produce a whole bunch of different enzymes. The main workers of the enzymes that are going to digest your food, they package them in vesicles. So again, this is Golgi processing. Enzymes that are packaged into the Golgi and often very tightly packed, so they're in dense looking granules under an electron microscope. They include lipases that break down fats, proteases that break down proteins, nucleases that break down RNA or DNA, and carbohydrate degrading enzymes. So a wide range of enzymes, and they're being secreted from secretory cells. And then I have a mistake in my video. I drew this sodium bicarbonate transporter moving the sodium and bicarbonate here into the lumen. But if you think about it, that's a secondary active transporter. It won't go that way. What actually happens is that it's this way. The sodium and the bicarbonate are transported this way. So let me go ahead and draw that in. It's a sodium and bicarbonate transporter that go in here, and then there are a bunch of different transporters that would allow the sodium bicarbonate out on the other side. We could have a bicarbonate transporter alone. We could use the sodium-potassium pump. And the actual one is slightly different, but there's a range of sodium. There's a range of possibilities that would move the sodium in. So I'll accept this because on an exam I would accept this because that's what I showed you. But I would, if I asked you a question that said, would that actually work? I'd want you to go, no. It would be driving, the sodium would be driving them this way rather than the other way. So, but if you put the sodium and bicarbonate transporter on this side of the cell, you can accumulate bicarbonate, which you can get out through a passive transporter, or you could use a chloride bicarbonate exchanger, a very common one, and then sodium you could move out with the sodium-potassium pump. And then all of these go down to the intestine. So the sodium bicarbonate, that is going to neutralize acid, and all the different enzymes will break down things in your food. Sketch showing the small intestine and how major nutrients are absorbed. So this is a cross-section of the small intestine. There'd be circular muscles around here and longitudinal muscles. So here's circular muscles running in a circle around here. There'd be longitudinal muscles. And these are constantly churning to mix your food. So squeezing and relaxing, squeezing and relaxing. And then there's folds, and then folds on the folds, and then microvilli on those. So zooming in, we see these villi. And the villi on them have all these little cells, and each of those cells have microvilli. Down at the base of these folds, there are stem cells that keep moving up and replacing cells that die up here. There are mucus secreting cells, and we'll come back to how the mucus is secreted, but the mucus is in two forms. It can be tethered to the membrane, or it can be free in untethered but sticking to itself in a mesh. Sodium bicarbonate is produced in mechanisms we just talked about in the pancreas. And then how are food particles absorbed? The same secondary active transporters we talked about over and over, sodium and glucose together, sodium and amino acid. So these same kinds of secondary active transporters. There are also passive transporters that pick up some food as well. But these will represent the kinds of mechanisms we're talking about for glucose and amino acids. And they're also transporters that will move things like phosphate and iron and calcium and any other nutrient or ion or whatever is needed. So this sketches how water is reabsorbed in the large intestine. Again, I would just want you to come up with some mechanism that works. You don't necessarily have to memorize this one, although I'd want you to understand it if I showed it, and you would need some mechanisms that work, and this one works. So again, this is based all on the sodium-potassium pump. We're creating high sodium outside and low sodium inside. Here is the gut lumen, so this is the ECF, where sodium-potassium pump is acting. Here's the gut lumen, so here is the inside of the gut in this large intestine. How do we get water back so that we don't always have diarrhea and so that we don't keep losing the water and the electrolytes? So we want ultimately to recover the salt, the sodium, and the chloride. And we have waste products, and we also want to recover the water. And a waste for us is carbon dioxide. So one way to recover the sodium is by a sodium proton exchanger. This will move in by secondary active transport because of the low sodium, and then of course it'll be pumped out. Where do you get the protons? Well, carbon dioxide is a waste product, so this familiar reaction catalyzed by carbonic anhydrase, water plus carbon dioxide. If we have carbonic anhydrase, this happens relatively rapidly to make carbonic acid. Some of the carbonic acid will dissociate to produce a proton and bicarbonate. If we have a transporter that moves that proton out, so that's the source, then some more of the bicarbonate will dissociate. A second transporter exchanges the bicarbonate for a chloride, and so we're recovering now sodium over here and chloride over here, and we're getting rid of what is a waste product. Carbon dioxide, which of course we could exhale, but if we can recover chlorides, valuable chlorides and sodiums, why not get rid of it in the gut as bicarbonate and carbonic acid? Now once in the gut, most of these will actually reform into carbonic acid. It's a weak acid. So I think at physiological pHs in the gut, maybe 19 out of 20 of them would be in this form, and only about 1 out of 20 would be in this form. But if we keep moving in protons and bicarbonate, we're going to keep H2CO3, and that in the process of letting these go across, we are recovering all these ions. There's also a sodium chloride cotransporter, another secondary active transporter. So here's another way to get sodium across, moving in because sodium is in low concentration in the cell, and all of these chlorides then move across because of the high concentration of chloride and the negative charge inside the cell. We drive some out. Finally, a sketch summarizing mucus production in the stomach and small intestine. So here is a cell, and not shown is the Golgi, but we'd have rough ER, ribosomes on, so ribosomes that are associated with endoplasmic reticulum, and they would be, oops, here's the messenger RNA going through the amino acids chain. The protein goes into the endoplasmic reticulum. It's then moved through the endoplasmic reticulum to the Golgi apparatus, and then vesicles from there are secreted. In the Golgi, the protein, the mucin proteins, are given all these side chains of sugars, and that's what makes them sticky to each other, and it also makes them absorb water. So here are the side chains of sugars. They can be in two forms. One form of mucin is just secreted with all these free sticky mucus strands with all the little side chains on them. In the other form, they're actually tethered. The mucin is embedded in the membrane. And so in the one form, this one, we just get free mucus in these sticky mesh. And if it's the membrane-bound version, then all the mucuses are stuck. And so here is a mucus stuck, and those can be shed. If a bacterium penetrates in that close to the cell, the bacterium is sensed there, and that triggers a second messenger, a local pathway, that causes the mucus to be shed, carrying away the bacterium. So it's one of your defenses. And then the mucus is also providing a barrier for acids and pepsin. In your stomach, for example, you have pH 3.0 on the other side of the mucus barrier, but right near the cell, the pH is closer to neutral. And so the cell is trapped. Now, is this barrier perfect? Absolutely not. Some acid can get through, and these cells in the stomach have a short lifespan of just a few days. The same events are happening in the intestine, so I won't go through them all over again. Clearly the acid and, in the stomach, clearly the acid, pepsinogen, and acid and pepsinogen need to get through the mucus layer in some way, and so there must be small thoroughfares through which the acid and the pepsinogen can get in the stomach. Once on this side, it's acid, and there's pepsin, a very harsh environment, so pH 3 and lots of that, lots of pepsin.